So I'm going to be talking about phylogenetic comparative methods today. And my job really is to be linking, what I'm seeing as my job, is to be linking the two halves of the course a little bit. So talking about how we do phylogenetic comparative methods and what that means in terms of the microevolutionary models underlying them and how we interpret the results. This isn't doing anything. I'm going to go old school here. Okay. So I wanted to start with just a quick review. So Joe, as I understand it, introduced the comparative methods to you yesterday in contrast a little bit. So what I want to do is just start with a quick review to make sure we're all on the same page, and then move into always thinking about the evolutionary underpinnings of what we're doing. Okay, so why do we do comparative studies? We do them for lots of reasons. I mean, I think the main one is really because people want to draw a general conclusion. So they want to analyze data from a broad group of taxa and come up with some general statements. But the reason that people do phylogenetic analyses, or why put a phylogeny into that, I think breaks down into, well, there are three main ones that I hear that people are using. The first one is a pure statistical reason. It's about sample size, right? So you go out and you measure a whole bunch of species, and you want to do a statistical analysis. You need to know what your sample size is. And at first you might think that your sample size is the number of species you have, but because those species are related to each other by a phylogeny, we know that that's not quite right. So here I've got 8 taxa, and we might think that our sample size is 8, but if you look at the phylogeny, you might start thinking that, well, maybe these only count as 1. Right, because they're linked to each other. And so maybe our sample size is more like 4. And because all phylogenies come together at the root, right, maybe we only have 2, or maybe we only have 1. The truth is really, it's somewhere in between all of that. And the only way to really know what our sample size is to put a phylogeny in the analysis. Okay, so that's one reason. A second reason, and I want to tell you, I will put these slides up on the WordPress site afterwards, in case you don't, so you don't have to write down everything. The second reason that people do it is to detect patterns. So imagine that I've gone out and I've measured a bunch of taxa, and these are the data points. So each data point here is a different species that I've measured. And I'm looking for a relationship between two traits, and these could be anything. It could be, in my case, it's usually something like body size and then some behavioral measure. On the other side, so it might be home range size and body size. And I want to know if there's a relationship between these two, okay? And I plotted them here, and I see nothing, right? No evidence of a relationship here at all. It looks like they're independent of each other. But if I then go and look at the data a little bit more closely, and I notice that these points were all measured from different species of fish, and these are all different species of lizards, I might come to a completely different conclusion. So now I find evidence for a strong positive relationship between these two variables, but it really depends if I'm talking about lizards or fish, right? So, and the only way to detect that pattern would be to incorporate that taxonomic information, or better yet, a phylogeny that would give me that information. Okay, so those are two reasons that I actually hear most often from empirical biologists who want to apply comparative methods. But I think the reason that I do comparative methods, and probably most of you want to, given that you're here in a course called Evolutionary Quantitative Genetics, is because you want to infer something about evolution, right? You want to understand something about evolution. And comparative data have this powerful potential to give you insight into evolution in ways that you can't get in any other way, right? So if you imagine a phenotype at each generation of time, so this might be body size, for example, and at each generation it might increase or decrease, and it goes over time, and this is evolution along a phylogeny. So every once in a while you have a speciation event, and evolution continues, but it splits along those two lineages. And what you end up with at the end is a set of comparative data. And if you have those comparative data and you have a phylogeny, technically you can write down equations that will work your way back to infer all sorts of things that happen at the microevolutionary level. So you could infer selection, you can infer the drift, you can infer the power of different environments. So it's this this promise of being able to gain this insight into something that you may not be able to get in other ways. Okay. So what I thought we'd do today is to walk through, this is just an outline of where I want to go to keep us on track here. So I want to talk about expanding assumptions, extending questions, and then I'm going to go off on a couple of newer and better ways of doing things. But to get started, I want to give you an example, just because I'm an empirical biologist at heart. I need an example to keep us on target here. And the system I work in, so my favorite example are Scloporus lizards and their head bob displays. So these are lizards that are found throughout North America. There's about 100 different species of them, and they communicate in lots of different ways, but I'm going to talk about two of them. One of them is a motion display. I'm going to show you here. You see the lizard right here? Keep your eyes on the lizard for a minute. Yeah, it wasn't. Yeah, it wasn't quite connected. Yeah, but I'm okay. I'm better with that. I'll do it again. Okay, everybody watching the lizard? Okay, so that's a head bob display. That's a real lizard in the field doing this head bob display in Arizona. And that's how we describe it way up there. So that's the x-axis there is time, and the y-axis is how high it's raising its head over time. And they do that display, that particular sequence, not just one bob, but that particular sequence, as they roam their territories. So at particular perches, they'll stop and they'll produce the display, and then they'll move on and they'll do it again. So that's one variable that I want to look at. The other variable that I want to look at is over here, are chemical secretions. So Scloporus, the genus name is means pores in the thighs, right? And so they're named after these pores that they have. This is a lizard turned on its back, and along its hind leg here, you can see these pores that produce a chemical secretion. So it's a second communicative display, second way that they communicate. And they're scent marks, so they actually do it in the same way that they do head bob displays. They wander around their territories and they produce these chemical secretions, and they lay them down on perches. So we have these two different ways of communicating. They're really pretty different from a behavioral or a physiological perspective. I want to know if they've evolved together. Okay? And since I do comparative methods for a living most of the time, what we've done is a comparative analysis on this. This is just a few species so that they fit on a slide. We have 39, data from 39 of them here. And this is a collaborative project, so these are some of the people that are involved in it. These are the head bob displays patterns. So this is the one I just showed you. This is a Virgatus was producing that display. And you can see that other species give really different displays. There's some variation within species, but across species it's way bigger. And in addition, we have counts of these pores on the thighs of the lizard. So again, it's a species typical thing. There is some variation within the species, but mostly the variation is across. And the question is, are these things related to each other? Okay? So the first thing we did was just plot them, and here they are. Each data point here is a different species, right? And what we see is, well, not much of anything, right? When you first look at it, it doesn't look like there's much of a relationship. But we can calculate a correlation coefficient. Yeah?
Could you use the microphone, please?
Oh.
In the diagram of the motion, what the spots mean? Seconds or?
No, it's number of head bobs per display.
Oh,
so we
okay.
just counted the number of head bobs per display. We actually tried about 50 different things that you can measure off of those, but what I'm giving you is the strongest pattern that we found, and an example of it that we published here. So it's just counting. That would be a 5. Okay, so not much of a relationship here. We can do standard statistics on this, right? So this is a Pearson correlation coefficient. It's a negative 0.3. So there's a slight negative. You can kind of look at it there. Actually, if I do it as a regression, and fit a line, you can see it a little more clearly. A negative relationship there. Okay, so what I want to do, though, is talk about what we're assuming when we do this, and what does it mean from an evolutionary perspective, okay? Everybody with me on this? Okay. So here, I'm building into a regression framework, by the way, so I just moved from correlations to regressions. I hope you don't mind too much. This is a basic regression, and the assumptions that come off of it. So the y equals beta x, the y is the number of head bobs per display, the x is the number of femoral pores, right? And we're assuming that the residuals of this model, the error term, are normally distributed with a mean of 0 and a constant variance, right? Yeah, before I go there, actually, let me ask you guys, what are we assuming here about evolution? Go ahead, Minzy. Yeah, I'm sorry, you have to use you're going to be running here a little bit.
Normally distributed?
It's normally distributed, yeah, but what does that mean in terms of the variables? So, you're saying the variables are normally distributed?
Yeah.
Right, so my number of head bobs per display is normally distributed, right? Okay, what else are we assuming?
Independent, so this is like assuming a perfect polytomy, right?
Right, so the star phylogeny,
Yeah.
right? This was Joe's depiction in his 1985 paper of the phylogeny that would produce independence. Okay. What else are we assuming? Anything else? So we're also assuming homoscedasticity, right? So that the variances are equal across these species. So it's not only that they're independent, that they arose from the same place, but they've each been evolving for the same amount of time coming off of that, right? Okay. And that the process of evolution has been similar, right? Generating the same amount of variation across that. So the question I want to be asking today is, what sort of evolution, I mean, thinking as biologists here, what sort of evolution would generate these kinds of data, right? And so for this, what I'm calling tips, just for historical reasons, referring to the tips of the phylogeny. So for this, what we have, some of the things that could create this sort of pattern are, for example, strong and rapid selection. If you have really strong selection that's pushing the trait in a direction or another, that you completely erases history, right? Or any other force like that that's going to erase the effect of the phylogeny. Okay. So we're not arguing that the phylogeny actually looks like this. We're arguing that the expected amount of dependence generated by the phylogeny looks like this. Okay. Okay, and I wanted to point out that another point here is that sigma squared I in the residual term there is a matrix, right? And it's a matrix that has the species on one side and the species along the top, so on the rows and the columns. And the entries here are the expected dependence between those taxa, right? And this is an identity matrix with the sigma squared along the diagonals. Okay. And that's the basic form that we use for a non-phylogenetic analysis. It's a really unlikely set of assumptions, right? It seems unlikely for many biologists when you're thinking about your own favorite system, and I really hope you can do that. Do you all have a favorite system in mind? Think about your data set and how you might, how those species live. And when I think about head bob displays and I think about femoral pores, they're not like this. Okay. So an alternative that we can do, I love this picture of you, Joe, by the way. And, so another way that that we can do it is the contrast approach that you heard about yesterday and tried out I understand, in the computer lab. So the basic idea, just to get us all on the same page here again, is that even though the species are not independent of each other, there are certain differences or contrasts on here that are independent. So we can generate a bunch of differences, A minus B, C minus D, that are independent of each other. And then if we take another step and assume Brownian motion as a model of how these species have evolved, we can prune the tree and we can generate a bunch of other contrasts here that will give us data that conform to the usual assumptions of regressions. So they'll be independent, they'll be homoscedastic, and they'll be normally distributed. Okay? Brownian motion was just an amazingly powerful idea, right? And it was a great idea to bring it into this context because it can do an awful lot. So in recent years, we've been going away from Brownian motion in lots of different ways. But to tell you the truth, I still always, almost always, when I'm done with an analysis, I go back and believe the Brownian motion and not necessarily the other complications. So it's something that, that we should hold on to. Okay, so back to the example. This was the TIPS line that I drew, so the non-phylogenetic line. If we do the same sort of analysis using contrasts, this is what happens. So we could get a correlation coefficient, it would be a negative 0.8, or we can fit the line, the red line here. And so what happens in this example, and the reason I use this example actually, is that you get a much steeper relationship when you use the contrast approach than when you use the non-phylogenetic approach. That is not the norm, okay? So normally it goes the other way. You have a significant result, you apply contrast, and it goes away. But in some situations, you get this beautiful uncovering of a relationship that wasn't there before. Okay? Okay, so I wanted to talk about Brownian motion for just a couple minutes. Back to what are we inferring? What are we assuming? What kind of evolution does Brownian motion describe well? What do you guys think? Since you talked about it yesterday, right?
So Brownian motion describes drift. So a random walk without selection.
Yeah, so you could use it to describe a trait that's evolving by random genetic drift, right? So no selection at all, drift. I really don't think my head bob displays are evolving by drift, but maybe they are, right? That's one possibility. Thank you. What else? Any other options? Imagine anything else? No? There are, oh, great.
That what happens in the past doesn't affect what happens afterwards?
Yeah, so it's, you're right. What happens in the past doesn't necessarily affect what comes afterwards. But what's important here in the Brownian motion thing is that things are following the tree. So there is some of the variability is determined by the fact that they're on a phylogeny, right? So it's not completely what happens in the future doesn't depend on the fact that there was a phylogeny and evolution happened. Okay, so we have random genetic drift. Did you have something else? No. Okay. Okay, random genetic drift, that's the first one we always come up with. I wanted to raise a couple of other ones. So the other ones that are often mentioned is fluctuating directional selection. So we could have directional selection. It just needs to change all the time, right? So the randomness doesn't need to be in just the phenotype. It could be that selection is pushing it in different directions every generation. You get a different type of selection acting on it, right? Or maybe every few generations. So as long as that selection is random, and that actually kind of fits my lizard situation. Another situation that could be happening here is constant directional selection. So if you have directional selection, but it's in one direction all the time through the life of the phylogeny, that won't change the the variability among species, right? It'll just change the mean, change where they are, but not the distribution of them. And so Brownian motion might work for that too. Okay. So I know yesterday you were talking about contrast in sort of a likelihood framework. I want to put it into the generalized least squares framework, the regression framework here. So it's the same model, right? We still have y equals beta x plus e, and we're still assuming something about the residuals. They're normally distributed. They have a mean of 0. But what we do is we put this V matrix over there in the variance, which incorporates the phylogeny. Okay. So in that V matrix, what we need are, it's not the phylogeny itself, but it's how much we expect the trait to change given that phylogeny and how evolution happens along that phylogeny. Okay. So again, we have a matrix. This is the V matrix, species going down the side and across the top. When you apply this in your computer exercise with your own data, one of the first things I do with my own data is actually write out a bunch of these by hand. And I know I'm old school. I don't like a lot of more modern things, but I like looking at them. It really does, it helps you check for errors, but it also helps you just think about how evolution happened. Okay, so in this case, the entries in the matrix are sigma squared TA, where TA is the amount of time that the species evolved together as an ancestor, as a common ancestor. And when you draw them out, this is just a hypothetical here, what you find is that there are blocks of things on the matrix, right? So if you look at enough tree matrices, this is what they tend to look like. You have these big blocks of zeros in places where you're going across different clades in the tree. So this clade is not related at all to that clade because we put it across the root. And so you'll get zeros in the matrix. And I just want to emphasize again that this matrix is not necessarily the phylogeny, right, by itself. It's an expected similarity matrix, how much we expect this particular phenotype to be dependent on each other, right? And we use, did I just lose this? Okay. And we use the phylogeny as a way to infer that, but we might use different things, right? We might make other evolutionary decisions to use some transformation on that matrix. Okay. So we talked about these different ways, these different reasons that Brownian motion might fit a phenotype particularly well, but it doesn't work for a lot of other reasons, right? A lot of other situations where Brownian motion might not be so good. Selection is a big part of that. So most forms of selection that you might think out there might not be fit well by a Brownian motion. And the other one that I'm going to be talking about later on is phenotypic plasticity that may or may not be well fit by a Brownian motion. Okay. So basically all comparative methods, whether we're doing a tip sort of analysis or a contrast sort of analysis, they all make evolutionary assumptions. And that's the take home from what we're doing here today, really, is that every time you analyze comparative data, you're making some evolutionary assumptions. They're assumptions on the one hand, so a problem, a nuisance that you have to deal with, but they're also a way of inferring evolution, because if you could estimate some of those things, you could learn something about evolution. Okay, so that was the first part about extending assumptions. I want to move now into expanding assumptions, and now I want to move to questions, extending the questions that we might ask. Because I think these are the two advances that we made with developing PGLS. So PGLS is phylogenetic generalized least squares. And it's something that people have been working on for a long time. A lot of different people have invested in this general approach and developing different tools that you can use under a PGLS sort of scenario. I attributed the initial idea to Graffin in '89. And the basic idea was just that we use a regression because then we can use the power of regression statistics. We know a lot in statistics about regression and how to do it. And it's a comfortable thing for most empirical biologists to use. Most people know how to run a regression in one way or another. And so this puts it into a framework that people can use more readily. And it also gives us a huge number of tools that we can access without having to build our own. software. Okay, so so we've talked a bit about how you set up the basic model, the y equals beta x plus e, so the different characters of the y's and the x, and then we have the assumption about the residuals in the model. And we've talked about changing this V matrix, or in TIPS using an identity matrix, or using something like Brownian motion to give us a different V matrix. Okay. So in terms of expanding the assumptions, so there are two things that we're going to be talking about, expanding assumptions and the questions. And in terms of expanding the assumptions, what we can do is move beyond Brownian motion and look at other sorts of assumptions that we might put in here. And that involves changing that V matrix in different ways. So Brownian motion was it's the idea of particles in a box, so they're moving randomly. The next step from that, from a statistical perspective, was to use an Ornstein-Uhlenbeck process, or an OU process. This expands Brownian motion by one parameter, basically by an alpha parameter. And it's called the rubber band process. So I usually think of it as particles in a box, and then you have a stick going through the box and a rubber band around the stick. And the phenotype is attached to the rubber band. Okay, so the idea is that the phenotype can move around randomly, but it's attached to this stick by the rubber band. So it can't get too far from the stick, right? If it goes really far away, the pressure to push it back is much stronger than if it just goes just a little bit of distance. There's a little bit of pressure, but not much. Okay? And the alpha parameter is the width of the rubber band. So it's the strength of that constraint. If you have a really wide rubber band, it'll be pulling you much faster towards that optimum. Okay. And this is a model that's been developed for a long time. Russ Landy talked about it in 1979 as a model of phenotypic evolution, something that we can describe how evolution happens. So it's a natural next step from Brownian motion. It's sort of Brownian motion with a constraint towards this optimum. I took this figure from Butler and King that shows you a little bit what happens to phenotypes over time under these two models. So this one's Brownian motion. You start at the root of the tree down here, and then these are species just evolving independently of each other. I don't think there's a tree in here. But under Brownian motion, what you do is they become more different from each other. Right? Little by little, they spread. And we've got two parameters here, but in Brownian motion, it's really all about the sigma parameter here. The alpha parameter is essentially 0, or is 0. So if you make the sigma parameter bigger, then you get a bigger spread here over time. And if you look at an OU process, it's a little bit different. Here we have the alpha parameter. And this has been set so the sigma is 1 in both cases. So it's comparable to the situation over here. Right? But what Butler and King added here was an optimum. This red line is an optimum. So the phenotypes are getting pulled towards this optimum. They started out somewhere else, but they're getting pulled down to this optimum by the OU process. So you're still generating some variability across species, but it's also pulling them towards this spot. And the difference between these two graphs is the magnitude of the alpha. So here we have a stronger alpha, a stronger pull. Okay? And you can see that if the alpha was really strong, the species would go essentially instantaneously to the optimum, and it erases history. Right? So if there were a phylogeny under here, the species have just jumped away from it, from where they were before. Okay. So what we do with PGLS is that we're changing, again, this V matrix. That's where the phylogeny goes in Brownian motion or in OU. We just use a different formula here that adds that alpha parameter. So again, this was the similarity matrix that we have for the BM model. when we do the OU model, it's a different formula that we use to generate the entries in the matrix, right? We still have a matrix, but the entries are different. So here we have an alpha parameter. It's exponential to alpha. And the T matrix is a little bit different. It's a divergence matrix rather than a similarity matrix. Okay. But the point here is that the power of PGLS really is that we have one matrix. You can plug anything you want to in that matrix and still get the thing to run. This is a danger as well as a power because if you plug in whatever you want to, you may not be able to interpret the results as they come out. Okay. Okay, so the OU matrix is an alternative, it's an expansion. What sort of phenotypes might evolve in a way that can be described by an OU matrix? Or what sort of evolutionary processes might correspond to a rubber band? Think. He's coming.
Wouldn't it just be selection? Because the adaptive peak is acting as a rubber band.
Yeah, selection towards an optimum, right? So some sort of stabilizing selection is what we usually think about. I wanted to point out, too, that you can think of the non-phylogenetic approach, again, as being a very strong sort of selection, so it's erasing history really quickly. And the Brownian motion version is you can imagine it as a random process, a random genetic drift sort of process. And so the alpha parameter that's going in between these is telling you roughly how strong that erasing of history is, right? If it's a very high level of alpha, you get a result that's the same as the non-phylogenetic approach. If you have a very small level of alpha, if it goes to 0, you get contrast. Okay. So the type of selection that might be described by an OU would be stabilizing selection, potentially. There's some, we can talk about whether this should be weak or strong, and whether there are other things that might also pull things towards an optimum. And I wanted to just say something again. So a lot of PGLS, a lot of the discussion about PGLS these days has been, well, actually for quite a while now, has been about transformation. So there are people who will argue very strongly that you should transform your data if the data are not normal, or if they don't, if the residuals don't look quite right for your analysis. You might want to log transform your data. And the point I wanted to make here was just that there are a lot of transformations you could do. You could do a square root transformation or something else, but I'm hoping that when you do that, you'll be thinking about the evolutionary consequences of doing that, right? So what does it mean if you have to square root your data? They're probably not Brownian motion in the same sort of way that Joe described in his 85 paper. Right. Let's come back to that. I just wanted to get the seed planted. Yep.
I wanted to come back to the selection in those two processes. I'm just thinking, could you come back to the graph with both Brownian motion and OU?
You mean this?
Yeah, that one.
Uh-huh.
So I was just wondering, under OU, if we had an optimum in the initial point, so where it started,
Uh-huh.
would
So
that
if
be
it started
just
here?
they start? Oh,
Talking about two different things. That's the red line is the optimum, not where you started.
yeah, but I mean, if the optimum was, because on the y-axis there we have a phenotype, whatever
Right.
phenotype, right? So the starting point is somewhere
Yeah, so
up
the
there in terms of the value of the trait. So if the optimum was right there, if the stabilizing selection was centered at
Right.
that phenotype, how that would we would
Yeah,
just have
so let's think about this as like body size, for example,
Mhm.
right? So in this example, we have a body size that's it starts out much larger than the optimum, right? So selection's pushing it towards a small body size, right? In this example. And I think the question you're asking is, what if it's already at the optimum, right? What is the OU process going to do
Mhm.
to it, right? So if it's already at the optimum, you can imagine, well, here, these are already at the optimum, right? And what would happen if you just continued this spread? So evolution continues, and you get some diversification happens, right? That diversification depends on the sigma parameter,
Mhm.
right? And the alpha parameter will keep it from spreading further,
Exactly. Yeah.
right? So it'll constrain it within a band,
Mhm.
essentially.
Okay. Okay. And then the other scenario under Brownian motion, but with directional selection, we would basically have this this forking process going to the up or down, so the whole fork would be tilted,
That's right.
right?
Yes.
Okay.
Yes.
Okay.
So if you had Brownian motion, but with constant directional selection, pushing it towards an optimum, you'd get this same sort of pattern, but it would be shifted, be on a slant.
Mm-hmm.
Okay. Let me go back to where we were. Okay. So I wanted to put it back in terms of this example to show you. So this is the same graph we had before. We have tips and we have a contrast approach here. If we do PGLS, what it does, again, is it gives us one more parameter, this alpha parameter. It sort of shifts us in between those two. Okay? And when we apply it in this case, what we get is something that looks more like the contrast result. So it's intermediate here, but more towards the contrast result, more towards the red line with an alpha of 4. The alpha value is scaled to the units on your tree, right? So just saying it's a 4 isn't telling you much of anything, except for that I scaled this on a tree, so a 4 is relatively small. Yeah.
Is there any requirement regarding because yesterday we saw that we're supposed to make the regression go through the origin. Is that a requirement here too, or why? And by the way, why is why is the line not going through the origin? So it's like a reason of why or how can we do that without breaking the assumptions of the model?
Yeah. So sort of wanting you to answer this, Joe, but do you want to try?
If the vertical scale and the horizontal scale were contrasts, it has to go through the origin. But this has been put back onto the original scale, which you can do, and offhand I don't even remember how, but you've done it.
Yes.
So the slope was calculated from the contrasts, and then it was kind of reinterpreted back onto the original graph.
Well, actually, what I did to set us up for the lab exercise is I ran it as a PGLS. I ran the contrast as a PGLS, so then you don't have to actually do any transformations. It just does it automatically for you.
As long as alpha is not 0, you have to enforce the contrast value to go through 0, 0, right?
No, it's what Joe was
I mean,
saying is it
not on
matters.
this scale, but on the
It's
contrast.
just a scaling effect. So when you're running contrast, you're rescaling the x and the y, right? And when you rescale them by contrast, you have to force it through the origin. But when you scale it at this level, you don't.
Yeah, I guess my question is because alpha describes like on the scale, either use the tip or use the contrast method, right? So if alpha is more towards the 0 or the tip value, so when you're just using the tip, Now, I guess you don't have contrast value.
Yeah, so when we're doing it in this PGLS framework, we're sort of getting around the algorithm of the contrast thing. So the way you do it is actually a little bit different, but the results are identical.
So in effect, the PGLS incorporates the transformation. It's as if it transformed to contrast, did the regression, and came back, except it's, if you do it more generally in this Ornstein-Uhlenbeck case, it's also including all of that machinery and doing what you would, instead of contrast, it's doing the more general OU machinery as well.
That's right.
And ends up giving you the plot on the original scale. If you were just, if it ended up at estimating alpha equals 0, the plot you'd get on the original scale would be the same as if you went to contrast, regressed, and came back.
So one of the nice things about PGLS is that it uses this linear model approach that allows you to do all sorts of different changes to the model, right, that way, that are easier to do than an algorithmic approach like the pruning.
Now that we're in this slide, I'm having a hard time thinking that head bobs being a discrete variable evolves, or you can fit a normal distribution or a Brownian motion to it. Could you please comment on why this works?
Yeah, well, and I'm glad you noticed that. I sort of leave that in the background hoping that somebody will bring it up. So it's not really normally distributed, right? I mean, probably not. It's a discrete ranked variable. So it does have some variability here. It's not just 0, 1, but the critical issue for PGLS, though, or for any GLS, is whether the residuals are normally distributed, right? Not whether the trait is just normally distributed, but whether the residuals are. And if you run this analysis and you look at the residuals, they're normally distributed. So that's why we went for it here. Often when you do this, that's not the case, and it won't look normal, and you'll be tempted to log transform it, right? Or to do a square root transformation, something else, right? And that's where I was saying that it's very important to think about it before you do that, because that really does fundamentally change how you think evolution is happening on the tree, right? But we were lucky here, didn't have to do that.
I want to argue a little with that and say it's important not to think about it. For a lot of traits that have a natural 0 point, like a length of something, it's much more frequent that things work out more normally distributed if you're on a log scale than if you're on the original scale. So I go so far as to tell students when they're learning about quantitative genetics from me that you should first of all take the logarithm and then only go back to the original scale if you have some reason to do so. So I'm kind of saying the opposite of what you're saying.
Yeah. So room to argue when we get to tomorrow. Okay. So that was talking about assumptions and that V matrix and how we can plug in different types of assumptions, particularly the OU there. And I want to move now to questions, because the other thing that you get out of putting it in this linear model framework is that you get flexibility over here in the mean structure of the model and how you set it up. So it's a regression, right? So we start out with thinking about 2 traits, but we could think about a whole bunch of different traits, right? We could have an X1, 2, 3, 4, 5, have as many as we want to. We could maybe not just have traits, we could put environments in there. We could put in, well, there are lots of things. You have that flexibility to move around. We talked about expanding the assumptions and how you could change the V matrix to be BM or OU or something else if you wanted to. Another thing you can do is to add within species variability or phylogenetic error in the phylogeny into that V matrix. And the last thing that you might do, because of this, well, there's lots of other things too, but another thing you might do is change the Y, the response variable, right? So instead of having a continuous response variable, you might want to do a logistic regression or a logit model of some kind, so that you can do categorical traits. Or you might want to go, wave at me if I'm not going live anymore, okay? You might want to go multivariate, right, and have a whole bunch of Ys that you want on there. So the general linear model approach opens the door to this, right, and allows you to think about doing all sorts of things you might do. And again, I don't know if Joe would disagree with this one, but I would say anytime you do those, it's an interesting exercise and something that you should pursue, but you should do it carefully and be thinking about the evolutionary consequences of what you're doing along the way. Because every time you add something, even just adding another x variable, there are consequences to the error structure and to the way that the phylogeny enters into the system that you should be thinking about. Yeah.
So, is it too weird to use this approach with genetic information, let's say genes or to like for doing orthologs, analogs analysis or stuff like that? Or, I mean, it looks like it might work, but I was wondering if there is some specific assumption that requires the tips to be species or something like that.
Right. So I'd like to think this is a great point for discussion too. We should be making a list. I don't know if you guys could do that of things that we can bring up in the discussion later on. But so there are people who have been doing all sorts of things, all of these things, and what you just suggested too. So looking at genes on the tips as the tips in different ways. I like to, again, separate out in my head the difference between the statistics and the evolution. So the statistics is very unlimited. You can do almost anything with these, right? It's the evolution that changes depending on what you do. So I think when you have genes as tips, you have to think through it differently.
I wonder if you can comment on when would you expand your assumption to let V include within species variation?
To use...
To let V include within species variation?
When would I do that? Yeah. I would do it whenever within species variation is large, right? Whenever it has likely to have an impact of one kind or another. Other people do it when there's an interesting question around the within species variability, right? Again, you can't just put it in. There are some things you need to think through. I'm gonna table that though because I'm gonna come back to it in a few slides. Okay.
I'm struggling with the transformation issue because I'm thinking, well, we let's say we transform our traits and then we model the evolution, but the thing that has been evolving is not the log. It is the actual phenotype. And when we think in terms of selection, and let's say we have body size or something, for what natural selection sees, the difference between 10 and 100, let's say, it's a completely different story than a difference between 1 and 2. So could you comment on that?
Yeah, well, actually what I want to do, I'm sorry to do this to you, but I want to table that. So I want to bring that up again in the discussion because I think it's an important point and one that we should talk more about, but I'd like to get I think everybody could comment on that. And I want to put a whole bunch of things in your heads and then we'll discuss. Okay, I wanted to give you just one quick example of this adding predictor variables. Let me go back. This is why I put this on the bottom here, and they went in the wrong direction. Is because I wanted to give you one specific example of this, just to get it in your head what you can do. And that's because in my lizards, I talked about motion and I talked about chemicals, but I had this picture up here, but I didn't mention that they also have this color. Right? They have a color signal. They have these bright blue belly patches. And evolutionarily, they've lost it a few times. So about 9 times in the clade, they've lost the blue belly patch and gone to a white. We think it's because of predator pressure potentially acting on the very conspicuous blue belly patch. So what I really wanted here was not just a correlation or a relationship between motion and chemicals. I wanted to know how the color went into it too. So I had this other x variable that I wanted to put in there. And because PGLS is a general linear model, it's easy enough to build an ANCOVA. So this is the way we would put in color. in our model here, right? And then we would fit two slopes. We would fit one slope to motion and chemicals for the blue-bellied species and one for the white-bellied species, and see if there's a difference between them. So we did that here. You can see the blue dots are the ones that have blue bellies, and the white dots are the ones that have lost that signal, right? And you can fit the two slopes. And what you find is that it's a steeper slope in the white-bellied species, that's the black line here, than in the blue-bellied species. And this made us think a little bit further because, so adding another x variable was not a huge deal, pretty straightforward using the PGLS framework. But it made us start thinking evolutionarily about what's happening here. So the basic relationship is that there's this trade-off between the two traits, right? So there's some species that tend to be more variable in the head bobs and others that are more variable, going to a higher level here in the head bobs and others that are going higher, more ephemeral pores. And that in the white species, so when they've lost the color signal, they're becoming even more specialized, even more one way or the other. And this made us start thinking about adaptive radiations and actually adaptive specialization. So you can imagine on a phylogeny that these are again the head bob displays, right? And these blue arrows are where they've lost the blue belly patch. And you can imagine that that the blue belly patch acts as sort of a key, not a key innovation, but a key loss. So when it's lost, that maybe it does something to the context in which the motion and the chemicals are evolving, so that it makes it even more likely that they'll go one way or another. Okay. So to look at that at the same time, this again is our initial ANCOVA model with the two slopes. What we did is we decided to borrow something that Brian O'Mara developed to look at rates of change also. And we fit a V matrix. So the V matrix, again, is where the phylogeny is. And we split up the phylogeny into areas where they have blue belly patches and areas where they have white belly patches, and estimated two different rate parameters in those at the same time. And again, you could have done this with a likelihood framework too, but I tend to think in terms of GLS. It was straightforward here. And what happens when you do that is you find that the variability in the white species has also decreased. Okay. So what we have is that the evolution of this color patch, when the color patch is lost, that you get this increased specialization in the other two signals. Okay. So I wanted to use that as an example just because I wanted to point out that because this is a general linear model, it gives us this flexibility to ask lots of different questions. We can talk about rates of divergence or amount of phylogenetic effect or estimating ancestral states in a framework that's relatively familiar, right, to a lot of people. A lot of people have been using it for other reasons because of that, right? So they've been developing their own questions. And I just wanted to list a couple of them here. People have been starting to incorporate fossils, looking at biogeography. I'm actually have a project going on character displacement that I'm really excited about now in sympatry. Co-speciation, so people trying to put trees together in different ways. And people have also been talking about communities. So on the tips, instead of having individual species, having communities of species that are evolving. So these are all different things that people have been talking about and working on developing PGLS sorts of ways of addressing that. And again, the point that I wanted to make that Joe's going to argue with me about later is that I think that they all have evolutionary implications. And I think what's really interesting, this is the interesting part, right? That's where we can learn something about evolution in a way that we didn't know before. Okay. I'm already running into discussion time here, but we've been talking a little bit, so I'm going to keep going. I'm just going to speed it up a little bit. Good. I just wanted to talk about two things. PGLS is a way to sort of if you're an empirical biologist, to try to fit all of these things into a framework that you're comfortable with. But it's also possible and better, usually, to think about evolution from first principles and to develop models that are designed explicitly for your situation, right? And there are a couple of places where that has shown itself to be particularly important to do. And the first one that I wanted to mention was in modeling selection. So this is Thomas Hansen. And we worked on PGLS together a long time ago. And while we were doing that, he was always complaining because he didn't like the basic idea that people were using contrasts to compare the relationship between a trait and the environment. Saying this is a very simplistic way to do it, and that we really think of evolution, if you think about it a little bit more, is more complicated than that, right? And so he developed this method to look at it, and what he said was that, so he came up with a different way of describing how evolution happens, and what he said was that evolution happens in a complex world, right? So we have selection acting on it. So these are horses that in one habitat they were browsing, and another habitat they were grazing. And that this difference in habitat has a big impact on their teeth, the morphology of their teeth. And in that if we really think about it, in this case, we think about it as being an essentially instantaneous effect. That selection can have that effect really quickly, right? And so we wouldn't expect it to be sort of randomly drifting along this phylogeny, the teeth shape to be randomly drifting along this phylogeny with selection having that instantaneous effect. But that what happens is that it doesn't have that instantaneous effect, or not completely, because the world is a complex place, and there are lots of selective pressures acting on horse teeth at the same time. And so, so the phenotype will not jump to another phenotype immediately because it's being pushed in different directions by these other phenotypes, by these other selective pressures. Sorry. So there's a complex selective regime. The other point he made was that this was sort of a transformational thing back in the day. Before that, when people were talking about parsimony on evolutionary changes on trees, and you wanted to look at the relationship between evolutionary changes on trees, so you'd map out the changes on one trait, and then you'd map out the changes in another, and you'd see if they were on the same branches. And what Thomas pointed out was that lack of change is very important. Right? So it's not just where the evolutionary changes happen, it's where they don't happen. That's critically important, and then we need to incorporate that into our model. So he developed this model so this is a phenotype, and we're predicting it with the amount of time that they spent in these different environments. So it uses an OU process. It puts the environment explicitly into the evolutionary thinking underlying the model, and the phylogeny shows up essentially twice. It's in the V matrix, but it's also in the times that the species have spent in each of the habitat. Okay. So it's sort of pulling making the evolutionary question more explicit and more realistic, I think, for this particular scenario. Okay, so this was my example of selection, and how if you think about selection a little bit, you think about it in your own particular situation, you can come up with something that's perhaps more informative. It tells you more about what happened. I also wanted to talk about phenotypic plasticity as a second direction that people have gone in, mostly because phenotypic plasticity has been such an exciting area of empirical research over the last couple decades. And there have been a lot of people who address it from a phylogenetic comparative method perspective too, going back to Jim Chevron's work on this in '85, actually. It's the same year that the contrast paper was published. So he had a comparative data set in primates, or they had a comparative data set in primates that they were working on, and what they decided to do was to partition the variation in the same way that we do in quantitative genetics. So thinking about the variability in a phenotype as being separable into a genetic component and an environmental component, and then we can do an interaction effect between them, just like we do in quantitative genetics, right? Except for here we're talking about a phylogenetic variance, right, and a non-phylogenetic, or what he called a specific variance. this was an attempt to get at a measure of phylogenetic effect. So how much does the phylogeny impact the variability in our data? How important is it relative to other things that might be there? And he did it using a spatial autocorrelation model. And the only thing I want to say about this really was that the advance, or the interesting contribution from my perspective at least, was that it puts the phylogeny in the mean structure, right? So it's no longer in the residuals, it's in the mean structure here. It's something specifically that we're trying to estimate. How important is the phylogeny as a predictor of variation? And it gives us this coefficient on the outside, this rho parameter, that explains the amount of variation explained by relatives. So it's analogous to a heritability, right, in quantitative genetics. So this is a link between quantitative genetics and phylogenetic comparative methods that's not through the, well, it's not cleanly, it's only analogous from the biological perspective, but it's trying to use some of the statistics in the same way in the two contexts. That's Mike Lynch up there. He's my husband, actually, and I don't know who took this picture of him, but I found it yesterday when I was Googling for him. I thought it looked nice. So he, in 91, Mike, well, he was reading Chevron's paper and thought that the quantitative genetic analogy was a really interesting one, but that the math was all wrong. He didn't like the spatial autocorrelation thing. And so he decided to use instead some basic quantitative genetics, and he went for the mixed model, or the animal model, in quantitative genetics. So he took the basic idea, right, that we're going to estimate a phylogenetic effect and a non-phylogenetic effect, an additive genetic, right, or a additive phylogenetic effect, and a specific effect. But using the machinery that we have already from quantitative genetics. So again, it's saying that the phylogeny is in the mean structure. And from a biological perspective, I think what we're interpreting, both in this case and in the spatial autocorrelation approach, what we're saying is that there's some sort of process, like a Brownian motion process, acting through the phylogeny down here, right? But that at each point in time, does that work? At each point in time, we're getting some non-phylogenetic effect that may be happening. I've drawn it here at the tips, but it could be anywhere. It's an amount of sort of phenotypic plasticity, something that's not going to be inherited on the phylogeny. This is an analogy because that specific effect, or the non-phylogenetic effect, may be genetically controlled, it may be genetically inherited, right? It's just not phylogenetically inherited, so it's not passed from ancestors to descendant taxa. But we can still estimate heritabilities for traits. Using this, they're just phylogenetic heritabilities instead of genetic ones. So that's the phylogenetic mixed model. I should mention, there are some big issues in actually implementing this, and so there's been quite a bit of discussion about how to calculate the statistics, both in quantitative genetics and in phylogenetic approaches. So there's a lot of that if you're interested. I wanted to put it, just give you an example here to tie up the lizards for you. So we've done it in 3 different ways. We can also apply the PMM, the mixed model, to this. And in this case, what we got was a correlation of 0. And this is probably a technical pathology, right? So these things are hard to estimate, and we only had 39 species in here. So it's a relatively small sample size. But you can still estimate the heritabilities. And here what we got was a 0.3. So these are heritabilities. They go from 0 to 1, right? 0.3 for head bobs and a 0.6 for the femoral pores. And so what I learned from this is that the phylogenetic effect, right? So the amount that the phylogeny tells us about variation in these traits is different for my 2 traits here, right? That one of them is following the phylogeny more closely than the other. And that difference actually probably explains the correlation being 0 and creating that somewhat of a pathology. But it was hugely informative to me as a lizard biologist to be thinking about these two traits as evolving at different rates or having different types of evolutionary processes underlying them, right? Something that you don't get from the other methods. Okay. So that those models, both the Chevron Dowden-Leutenegger paper and the phylogenetic mixed model, again, they're getting at phenotypic plasticity in one way or another. And there's been quite a bit of discussion lately about phenotypic plasticity and what it means and how we can estimate it. Did you talk about that at all? About within species variability, right?
Today I briefly mentioned that one could extend to within species variation. I didn't talk about plasticity specifically.
Okay. Yeah, and the only thing I wanted to say, and Liam's sitting there, the only thing I wanted to say was that a lot of the discussion it's really interesting right now. It's a very exciting area from an empirical perspective particularly, because we're thinking about what is phenotypic plasticity and what does it mean. And this is a breakdown from the Hansen and Bartoszek paper where they're pointing out that even if we just think about it a little bit, what you might split out biological variation. So this would be real phenotypic plasticity, right? So real reasons that individual species are not the same, or some of them are more variable than others. But then there's also measurement error in both of the traits or in all of the traits that you have on there. And that if you can incorporate those into your error term, into that V matrix, for example, in different ways. But when you do so, you have to think about the details a bit. So for example, whether the error is in the Y or the X matters. Here we have a regression coefficient in the X that you would want to incorporate into that V matrix if you're going to include within species variability or even just measurement error. This is really important for a lot of reasons, but partly it's important for prediction. And prediction is where a lot of people are going these days, wanting to use comparative methods as a way to predict different things. So one thing is to predict phenotypes of extinct taxa, and this is an example that Liam maybe will tell us about later. Also, people have been talking a lot about response to climate change and trying to predict where phenotypes are going to go in the future. And I think for all of these things, that getting a handle on within species variability or phenotypic plasticity and all of these different issues that go into that error term will be important. Okay. So that's all I wanted to say about all of those, but and I'm going to open the floor to questions and discussion here in just a second. Before I go there, I just wanted to make one comment. I wanted to put NSF up here. So I started doing a rotator position at NSF on Monday. So I've only been there for a few days, and I'm going to be there for a year, though. And I just wanted to tell you, in case you have any questions about anything related to that, come find me later. I might be able to answer them or at least direct you in the right place. Okay. So let's open the floor for discussion. Questions?
I don't think I quite understand the difference between regular heritability and phylogenetic heritability. Could you go over that a little bit more?
Sure. So the similarity is easier to explain than the difference, actually. So the similarity is in how they're calculated, right? So when we calculate a heritability, we're calculating the resemblance between relatives and how much that explains a phenotype, right? And the heritability is a proportion, right? It's telling us how much that is explained. So in the phylogenetic context, it's the same thing. We're saying how much does the resemblance between relatives explain the phenotype? But we're talking about phylogenetic relatedness instead of genetic relatedness, right? You could have phylogenetic relatedness that's not driven by genetic forces, right? Yeah. Joe looks like he wants to say something.
I just say that when you, I believe in Mike's paper, he used terms like heritability by analogy to the same calculation done in the animal model. But if you look at his model, what you'll see is there's the covariances of change, which is the same as there is in other papers, and that may or may not be related to additive genetic variants, depending on what mechanism is causing the change. And we've kind of all been talking about that. And then when you get down to the specific individual component for each species, that has the same structure as if it were one of the possible things that could explain it is within species phenotypic variability. And that contains both additive dominance and environmental effects. So in a sense, the heritability, formally speaking, biologically in that model, is inside that specific component, not in the part he's calling phylogenetic heritability. So you have to watch out for terms that are named because of analogy of the mathematics and where that term is actually appearing in the model, which I'd argue is in a slightly different place. So, I mean, his model, by the way, is equivalent to part of my work in 2008 on within species variability. If you make all the sample sizes for each species 1, you get exactly Mike's model. So they're completely consistent. And in my case, the additive genetic variance is inside, it's in the within species phenotypic variability.
I guess, so is it fair to say that the traditional heritability speaks to resemblance between relatives in kind of the traditional sense, and phylogenetic heritability is resemblance between related species, sort of? Is that kind of the analogy?
That's the analogy, right?
Okay. So they're both resemblance.
Yeah. It's instead of a genealogy, you have a phylogeny. Right?
Isn't there a need for cause-effect relation between Y's and X to avoid linear dependencies within the design matrix that may make it difficult to solve the models?
Yeah, so PGLS is a regression, right? So we normally think about a dependency between the X's and the Y's. So the X's are predicting the Y's, right? They're not equivalent. You can't shift them around. And I think the only place I touched on that was with the measurement error, right? That because we're predicting variability in the Y, if you want to include measurement error in the X, you have to do it in a different way, right? You have to include the parameters that you've estimated from that larger model in the X. So statistically, though, what amazes me about statistics, and what really, I was never going to do statistics for a living, right? I'm an empirical biologist, but what really grabbed my interest and made me excited about this was the similarity among these methods. So that you can, if you learn how to run a regression, it's the same thing as a t-test and an ANOVA and or a logistic analysis. So it's in a regression framework, you can run a Pearson product moment correlation, right, and have no dependency involved. You're just moving the statistics around, right? It depends on how you set it up and do the model. So I think PGLS is, or my preference would be for people to think of it in that way, that it's a general way to approach the calculation of these things, right? It's not a way to determine exactly what trait means what, right? That you have to do sort of along the side, thinking carefully about it.
I think it's a good moment to say people may think about stuff we talked about before and say, well, it's kind of in a maximum likelihood framework, but here Amelia's going to a different statistical criterion, least squares. But actually not, because phylogenetic least squares is also maximum likelihood. It's just set up in a particular way where you can add other effects a little more, more easily. Right. So it has some advantages, but it's not as if it's contradictory to maximum likelihood approaches.
And actually in the computer exercise that you'll be working through, I set it up so you would run a non-phylogenetic analysis and a contrast analysis and then a PGLS-OU analysis all using the same statistical machinery. So the contrast analysis in there will give you exactly, or should, I hope, give you exactly the same answers as running it through Joe's approach, right? But it looks different.
I have two questions about what kind of evolutionary processes the Brownian motion can describe. So you said it can be either fluctuating directional selection or constant directional selection. I wonder if this makes assumption that the strength of the selection is constant over time. Because if it's changing, then the variance is not proportional to time. Is that true?
Potentially, yes. So it's sort of the strength and the weakness of Brownian motion, right? If you try to pin it down too much, then it doesn't do what you want it to do. But I like to think of Brownian motion as being this random process that can describe a lot of different things. So it might describe a situation where you have selection changing in strength in a random way. But it wasn't. Brian's wanting to save me here.
And also remember the exercise we did yesterday with simulating Brownian motion, right? Where we had, okay. So remember the simulation we did yesterday? We had, a bunch of different distributions adding together to make Brownian motion. So you could have selection changing through time as long as it's, as long as it has a finite mean and variance, right? And a bunch of different processes are adding up. It would still be Brownian motion in the end, right? As long as it changes fast enough, right? We had regime, if you had a long period of one thing and a long period of another thing, it might not be Brownian motion yet. You have to add up a lot of things, but at that point it becomes.
Okay. And I'm also a little bit confused as when can you apply that to constant directional selection? Because if it's constant, where does the little wiggly come from? Is that phenotypic plasticity or something else?
Oh, because you always have some variability underlying it, right? You always have the drift acting underneath things.
Oh, so it's directional selection on top of drift, for example.
Constant directional selection just without any drift would just put everything at the same phenotype and you'd be stuck there, right? There'd be no variability to talk about.
Mm-hmm.
So yeah, it's constant directional selection on top of a drift.
I was just wondering, how do you go about choosing the alpha value? Is it biologically, is it based on information, or is it that you have about the system, or
Yeah.
can't even imagine?
Yeah, that's a great question. Usually we estimate it as part of the procedure, right? So you estimate it from the comparative data that you're doing it from. That never quite feels right to me, but it's what we do. And because it's the information you have there and available. The other thing you can do is try a bunch of them, right? So you can just fit a bunch of them and then see how the results are affected by that. And I actually set up the computer exercise so that you can do that. Run a bunch of different alphas and just see. What I find empirically that is interesting is more that range. That if I varied alpha across this range, that this is how it would impact the real question I'm after.
If you have any a priori information about whether you think that there's a directional selection happening, would that affect your choice, or is it supposed to be independent of knowing?
Yeah, you could choose an alpha and fit it if you wanted to. Sure. So in the empirical situation, usually, it would be so nice to have all of this information ahead of time, and we don't usually, actually Steve does sometimes, has all of this information in hand, but usually we don't know how much to expect these traits to be changing on the phylogeny. So my usual approach is to try a range of possibilities, right? So to try a range of alphas, to try the BM and the OU model, to try the PMM, right? Run all of these different things, and then to ask how do these differences inform me about the evolution of the phenotypes, which is what I'm really after in the end, right? And they all give you sort of a different piece of the information.
this isn't my field, so I have. So if you know something about the underlying biogeography or the change in the biogeography over time, longer periods of time, how does that does one incorporate that into the model, or how does one do that?
It's almost like you were planted there, because one of the things that I'm really excited about, just one of the reasons I'm at NSF right now in Washington, is because I wanted to spend some time at the Smithsonian working on a project about biogeography. So people have been trying to incorporate geography into these questions, and there are lots of examples out there now that you can borrow from how you do it. The issue for me really is about it's that the species mean, the individual species mean, no longer seems like a real unit, right? So it's hard for me to put when I look at the biogeography, I look at the map, and I find that some species are really widespread, and their phenotypes may be slightly different in different places, right? That it's continuous variation across that map, and not just one number doesn't capture that for me really. So I think we're starting to get to a point where people are looking at how you might look at the geography, look at climate variables, and how those might inform us. We have lots of databases available now that you can gather this information, and also to put fossil information in and paleontological climate information in there to try to change the story. So I think we've come a long ways from just doing contrasts on two traits and looking at a correlation or something, right? It really is a good time to be thinking more deeply about evolutionary questions.
Depending on what level you're thinking of biogeography, I think there are very interesting things to do with large-scale biogeography where there are different species in different regions. But Amelia was also partly alluding, I think, to biogeography within a species. And in fact, that's an area that needs a lot of development, and there are good possible future methods that aren't there yet. Of course, I can always point to something I've written, but about I think it was 2002, I had or 2001, I had a paper in a multi-author volume in which I suggested that if you had optimum selection towards peaks, that where the peaks depended on a local environmental variable and you had multiple populations with a pattern of migration between them, nowadays with coalescent and molecular coalescent methods, we can start to make estimates of the migration rates between those areas. And I concocted a simple way of correcting for the biogeography when trying to figure out, for example, how, say, temperature affected, whether the species was responding to different temperatures in different regions by having different phenotypic optima. However, the method is not one that's sort of out there to use, and I think we'll need a lot more development before it comes up. But there's really interesting stuff to do within species using optima models, using migration models, and using estimates of migration between regions, in addition to all the stuff people are now doing very actively in trying to incorporate biogeography into between species models. That's been an area of very active development the last few years.
Yeah, that was what I was actually thinking of. I had recently been to a talk on human biogeography, and they had been coming up with, it's a long time since I was in grad school, I guess, not that long, but new models that beat isolation by distance in terms of trying to describe the genetic variation that they were observing. And so I was kind of wondering if that's, that seems like a cool thing that could potentially get incorporated, especially in smaller groups, maybe?
Yeah, I think a lot of the phylogenetic comparative method world went towards more and more species, so really huge, because of the sample size issues, because we're better able to estimate things when we go to these really huge clades. But that's not the whole world, and there's another big chunk of people who are trying to go for fewer number of species, but to go deeper into the population structure underlying that and individual variability within that, geographic variability. Is there something
Oh, I
over
thought
here?
you were going to go. Okay.
I actually wanted to use that as a segue to bring us back to an issue that you brought up earlier, which is one of the reasons I find the really huge species analyses a little unsatisfying. Is that to do those using a PGLS sort of thing, you have to assume something across the whole phylogeny, right? And so for me, that almost always ends up being Brownian motion. So it's because you're going across such a large phylogeny, and that's the only thing that could model the same thing across all of those different clades. Well, we know there are a lot of different evolutionary processes, and they're probably different in these different sections, but only something very random will get us there. And so we can't get at the details of the evolutionary interpretation by doing that. We really have to go smaller because evolution happens in different ways in different organisms, right? So the segue here, though, is back to transformations and what to do about that. So what do you guys think? How do you feel about? You just heard a couple of different extremes here on the transformation issue. Where do you fall on that? And not even in a phylogenetic context, just in general. Do you transform your data? Right.
So I have perhaps a really naive question. So if you can use logistic regression, that is to extend your residual structure into binomial, then why can't you extend that to other distributions?
Oh, you can, sure.
So if that's true, then a lot of your data, you don't need to do transformation then.
Yeah,
you can
so
just
that's an
use
interesting perspective.
different.
So let me say just the you can, sure response was, sure, you can do that statistically. You can do anything you want to, pretty much, right? So you could use all sorts of other distributions to describe your data and to put it into a PGLS framework instead of a transformation.
I want to also point out a technical issue, which is that it's not a matter of sitting around saying, oh, should I use a log? Should I use a cosine? Should I use an inverse hyperbolic tangent? But there are families of transformations, and there are what formulas statisticians have used to come up with transformations that have a parameter. And by changing that parameter, you can go from no transformation through a log, through square roots, through a log. In other words, you have a parameterized family of transformations. One approach that can be very valuable is to estimate that parameter by, for example, maximum likelihood. So it's not just a matter of you sitting around saying, oh, I wonder what transformation I'll use, but you can put that into the machinery and have the transformation estimated.
Right. So that's essentially what we do with the alpha in PGLS, right? So there's also the issue of transforming the branch lengths on your tree, right? You can transform the data, you can transform the branch lengths on the tree. What do you think about that? Any differences of opinion there?
Comfortable transforming the data, the phenotypic data, because at the end of the day, what I'm interested in is the relationship between them, not necessarily the quantities themselves, but how they relate to each other. So it's like, sure, whatever. But in terms of changing the branch lengths, I think that will be kind of weird, because that's the temporal structure of our biological entities. And I guess that will change pretty much all the calculation, the time that will take for the Brownian motion to get to one point, and that will get, I guess that will get messier than just tweaking it.
I'm not familiar with transforming trees necessarily, but I know with transforming data, one thing that you strive for is that the transformations don't change the rank order. They're monotonic. So like if you transform a tree, hopefully it wouldn't change the relative position of, the different lengths or whatever. So I don't think it's a problem. It's just kind of rescaling things. It's not changing necessarily their relationship to one another. So I guess it doesn't really bother me too much.
Since are not quantities, they are just proportional to or idealized versions of, then scaling all the branch lengths, it's trivial, right? I mean, it won't change anything at all.
So let me just throw out there again the point that what we need in a comparative analysis is not the phylogeny, right? What we need is a description of how we expect the characters to be related to each other. Right? That may follow the phylogeny, but it might not.
And I also want to ask, before we talk about transformation, do we know how robust the method is to the violation of normality?
You can get almost any answer depending on how you transform those branch lengths on a tree. Right? It has a huge impact. And that's one of the things I set up for you in the computer exercise. If you get that far, is just trying it out and seeing some of the variability you can get. But it can have a very important impact. So it's not a trivial question here. This is a big deal.